So yeah, so 12.4, we're just looking at some of these genetic disorders we went over in 12.3. What is how do they originate? And essentially they usually come about in some sort of alteration in the chromosome number. So cells are supposed to have 46 chromosomes, 2 copies of each chromosome, or an actual alteration in like the actual structure of one of those chromosomes. Okay. And an interesting thing is like miscarriages, so like, a spontaneous abortion, they don't know like how many of those are caused by like changes in chromosomal structures, but that's kind of like a leading kind of theory in like, what would cause like a miscarriage for a woman who's pregnant. The idea would be like there was the body detected some sort of chromosomal alteration in that fetus that would then lead the body to like, hey, let's abort this pregnancy right now because this baby's not going to be viable, and we don't want to risk, our lives and bringing it into the world and stuff like that. So I don't know if that brings maybe some, not that any of you have had a miscarriage, I'm assuming, but I don't know. That's just kind of an interesting thing to kind of like keep in mind. Anyways, what is interesting is plants, they can tolerate these genetic changes better than animals. And why would that be? Like, what do you think? It's kind of something your book just sort of says. They don't really talk much about it, but any thoughts? Think of the structure of plants. Like in 7th grade, like I taught them like the anatomy of plants. We didn't really go over it here. Versus like anatomy and physiology here, if you're in that class now. Like compare the two. Most of you. Basically, we're a lot more complicated than plants, right? We have way more. Yeah, we are. Yeah. Or maybe a better way to say it is we have a lot more different kinds of cells, more complex organ systems where if you have any sort of genetic change, that's going to complicate that kind of delicate sort of dance going on in the body where all these different tissue types and organ systems are communicating together. That would be the best guess we have for why plants can tolerate genetic changes better than we can. So meaning plants can like have like they could have multiple, so many different copies of all their chromosomes, and it's just fine. The plant's just doing its thing. It doesn't really affect it too much. Okay, there's a type of chromosomal abnormality called a non-disjunction. So this involves your homologous chromosomes. So what happens is, if I jump to the picture of it, we have meiosis 1 where we separate the homologous chromosomes. So the right picture would be like how it would be showing 2 cells. Here would be these chromosomes didn't separate properly. So meaning, I see. Never mind. Sorry. This is meiosis. So you're going from meiosis 1 to meiosis 2. So here, notice how because these chromosomes all travel together, this cell, whenever you separate the sister chromatids, you have 3 copies of each of the chromatids. You're supposed to only have 2, whereas this cell only has 1 copy. Okay, and then over here you could have non-disjunction happening in during meiosis 2 when you separate the sister chromatids, and a similar idea would happen where you're going to end up having an extra copy of every single chromosome number. Okay, so in other words, you could have non-disjunction of the homologous chromosomes, so during meiosis 1, or you could have non-disjunction of the sister chromatids during meiosis 2 when they don't separate properly. Okay? End result is you have 1 more extra chromosome or 1 fewer chromosome. Either thing is an issue. Okay. Let me go back and make sure I started. Yep. So, somewhat fairly simple idea. Then there's something called aneuploidy. So whenever you have fertilization of the gametes, that's just like describing whenever non-disjunction has actually occurred. So if you're somebody like if I jump over here, monosomic would be, so within like abnormal chromosome numbers, if you're a zygote that has only 1 copy of a certain chromosome, meaning you're like one of these cells, like the N-1, that they lost a chromosome copy, we call you monosomic. Whereas if you're a zygote that got an extra chromosome, so you're the N+1, that's trisomic. So these would go underneath aneuploidy. Those would both be examples of aneuploidy. Okay? And then polyploidy would be where you actually got like monosomic and trisomic, they're involving like individual chromosomes. Polyploidy is talking about you got a literal additional copy of all of your chromosomes, like the entire chromosome set, which is kind of wild. So like if you're an organism that has, you're supposed to be the diploid number, but now you have 3 copies, you're 3 in. Your triploid, we would call that triploidy. So he's kind of added a Y there. If you have 4 copies of all the chromosomes, you're tetraploidy. And for a similar reason we talked about earlier, that's more common in plants. Plants can have like, I think there's some plant species that have like 8 copies of all the chromosomes. And some animals, but like animals, begin because of how complicated we are, we can't handle just like, you just got an extra set of all of your chromosomes, you're probably not going to survive to like have offspring and everything. Why could this be beneficial for plants to be polyploidy, to have it like get extra sets of all their chromosomes? Thinking about like evolution, how could that be beneficial? So the idea would be if you have multiple sets of chromosomes, one set of those chromosomes could be undergoing evolution. They could be getting different changes in their structure that could then provide like variation in the environment, whereas one set of chromosomes could just be could stay the same and allow the plant to survive. And so that could be like the idea would be that would be like a way that plants could evolve in a different species. That an entire chromosome set, one of those chromosomes could start like changing structure and shape, but it won't overall change the actual functioning of the plant. It won't like prevent the plant from dying or something like that. Okay. Yeah, and that's what this last bullet point is trying to say. Okay. Now getting into, so that was going over like how you could change overall chromosome number. So now we're looking at, well, how could you change like an individual chromosome? So there's 4 different types you need to know for the AP exam. There's a deletion where you remove a part of the chromosome, a duplication where you literally like make extra copies of genes, inversion where you like flip it, and then translocation where you move one part of a chromosome to another chromosome. So let's walk through those. So deletion, like these letters would represent genes. So like here you'd have letters A through H genes. A deletion would be you just removed a gene. Like here they removed gene D. Okay. Duplication would be you duplicated an entire segment. So it could be one gene, it could be multiple genes on a chromosome. So here there's a duplication of genes B and C. So now you have BC, BC. Okay. Inversion would be like if you invert something, like you're like flipping it over. That's essentially what's happening. Like so you change the order of the genes on the chromosome. So you had B, C, D, but now there's an inversion. Now it went D, C, B. Okay. Now this one's kind of interesting because this is a little bit more livable, I guess you could say. What could happen here, we're going to talk in the next unit about gene expression, by changing, because you didn't actually change genetic information. We didn't technically lose genetic information. You have all the same copies of the genes, but you're ended up losing, see, have fun. You're losing by gene D taking over gene B's position. This could change how often we end up using gene D compared to gene B. If that makes sense. And I'll explain more of that in Unit 6, how exactly that works. And then translocation, that's where you're literally moving genes from like one chromosome to another. So here you have A, B, and then M and O, and they just swapped chromosomes. So now M and O is over here, and A, B is over here. Within translocation, this would be this one specifically is an example of a non-homologous switch because these are 2 different chromosomes. They're not homologs. They're not the same chromosome, same copy. There could also be a homologous switch where you had, if they were both like say chromosome 2, like you had genes switching from one. Basically, what happens in crossing over would be a homologous translocation. It's a similar idea. Okay. Yeah. So if you're looking at like kind of like a human diploid organism, if there's if you have a large deletion of genes, have fun. That's typically going to be a lethal genetic change for you. Because again, you're losing entire like genes. That's obviously going to be harmful. Duplications and translocations, they're not as bad as deletions, but they're typically going to be harmful. Like they're not going to be good things to happen because you're changing. Duplications, you're adding more genes, which can essentially lead to like you're making more of a protein than you need to make, which can cause issues. And then translocation, I was mentioning that where like that can change how often you use certain genes, which can be harmful. Inversion would be like the most like livable one, where like just by flipping the ordering of it, that can. I'm sorry, I said it backwards. The translocation, that's not altering the gene expression. It's the inversion where you just flip the location of them. Inversion will change the expression of those genes. So if I go back to that picture, here, like if you swap D and B, this is what will change the gene expression. How often you use D versus B. Okay. So yeah, said at the beginning of class, genetic disorders, like you change chromosome number or structure that leads to disorder. It's kind of self-explanatory there. An example now, technically in the like if you look at the AP exam like kind of objectives, they don't say that you have to know like the different genetic disorders. I do feel like you should remember Down syndrome. Down syndrome, they call it trisomy 21. So I mentioned that word. I don't know if it's trisomy or trisomony or if it's kind of tomato, but anyways, tri321 referring to chromosome 21. I think I would recommend that you just kind of keep that in mind. It's chromosome 21. I think it's easy to remember because of everybody's favorite rapper, right? 21 Savage. Yeah. Anyways, I don't know. I doubt they're gonna ask you like, which genetic change leads to Down syndrome on a multiple choice. It'd be more of like probably a free response question where they would like give you a narrative to read and they would ask you questions about, generally speaking, genetic disorders. But the idea here is by having an additional copy of chromosome 21, you're making, and I said this earlier, you end up having that extra chromosome copy, you end up making too much of certain proteins. It's almost like too many cooks in the kitchen, or like traffic jams, when there's too many cars on the road. That's what can end up happening in this, and that's kind of essentially that idea is what causes sort of the general Down syndrome characteristics. Just all those that extra chromosome copy making too much of the proteins. It affects about 1 every 830 children, so it's decently common. I think most of you probably either know or you've interacted with somebody with Down syndrome. It increases with the age of the mother, and they're not exactly sure why that's happening. Something about like the older the female's eggs get. I know especially as you start getting to like late 30s, like early 40s, if you're a female. Something with the eggs, like it just leads to a more likely chance that you can get an extra copy of the chromosome 21. Luke, yeah.
I think the reason why is because as you get older, your spindle fibers during meiosis become weaker. So
okay.
it's not disruption as well.
Yeah, that makes some sense. Yeah, Luke was saying the spindle fibers, like the idea they get a little bit weaker as like the female gets older in the eggs, and so the spindle fibers are in charge of actually distributing the chromosomes, right, during meiosis, and so that could end up leading to you a mess up happening where you get an extra copy of chromosome 21, and then what that would mean is if one egg got an extra copy of chromosome 21, another egg got one fewer chromosome 21. So there you go, and that's what kind of getting into what Luke was trying to say, this last bullet point. Okay. Now, within aneuploid conditions, if you change the sex chromosome, so like like XX for females, XY for males, that typically has less of an impact on the actual phenotype, the actual like how the organism behaves, than if it's the autosomes. Those are chromosomes 1 through 22. So, for example, there's something called Klinefelter syndrome where a male gets an extra X chromosome, they're XXY. And typically males that have that, they tend to have more like female characteristics. So like maybe like the hips are a little bit wider. They have a little more like breast development because of that extra X chromosome, making more of those gene products. There's another one where males end up getting an extra Y chromosome, so about 1 every 1,000. And this one's interesting because it doesn't really, they kind of appear normal, more or less, like having the extra Y chromosome. It doesn't really change things too much. And similarly for females who have trisomy X, they call it, they have 3 X chromosomes. There isn't really, like, the females are maybe like slightly taller than average, but it doesn't really affect them too much. So again, just this idea that if you have aneuploidy with these sex chromosomes, it isn't as big of a deal compared to the autosomes. Okay. Oh, and then there's monosomy X, or monosomy X, called Turner syndrome. So it's in females, right? Because there's no Y chromosome. And this usually means that they're just sterile. So, I mean, which, not that that isn't a big deal, but they, like, other than that, that's like the main kind of feature that happens. Like, it's not as if there's really other physical features that you would see. Like, you wouldn't necessarily be able, it's not like with Down syndrome, you can, like, kind of, I think you typically can, like, look at someone, you can kind of tell if they have Down syndrome. Monosomy X isn't quite as obvious that, like, oh, clearly you only have 1 copy of your X chromosome or something. And this would be the only viable monostomy in humans. So meaning this is the only situation that we know of where you can have only 1 copy of all your chromosome pairs and you're just essentially a normal human, other than just being sterile, where you can't have offspring. Okay. Another interesting one is Cri du chat. I think it's French. I don't know. Cry of the cat. This is where you actually have like a deletion of chromosome. Anybody notice it? Cry du chat. Say Cri du chat. Cri du chat. Yeah. Anyways, Cry of the cat. The name of it, which sounds kind of goofy because you're like, what? The idea this, like, where this name came from is babies that have this, when they're something about missing that portion of chromosome 5, they end up being born with this like cat-like cry, and so that's where the name comes from. They also will have like some pretty severe intellectual disabilities from missing that part of the chromosome, and they typically end up dying in infancy or early childhood. So it's not something that gets passed on really, because they're going to be sterile. Certain cancers, so chronic myelogenous leukemia, certain kind of leukemia would be caused by a translocation of chromosomes. So again, for the AP exam, you don't have to memorize these specific disorders. It's just something to keep in mind, just as examples of how how like changes in chromosome number or structure can lead to a genetic disorder. Okay. And here's the example of CML where comparing chromosome 9 and 22. So what happens is they end up doing a translocation where they swap portions. This would be an example of a non-homologous chromosome translocation, because these are chromosome 9 and 22. They're not the same chromosome. Any questions? Okay. So yeah, the rest.